This training video guides you through the steps to successfully conduct a test using Neogen's Reveal Q for DОН and the AccuScan GOLD reader. This presentation serves only as a visual guide to the written materials supplied with the test kit. To ensure accuracy in the performance of this test, please read and follow the test's written instructions in their entirety. Each kit contains enough sets to test 25 samples. The kit includes test strips, Reveal sample cups, sample dilution cups, sample diluent, and the QR code. To conduct the test, you also need the following: sample collection cups with lids, an AgriGrind or bun grinder, a scale capable of weighing 5 to 50 grams, accurate to plus or minus 1/10 gram, a timer, a Reveal sample cup rack, the Reveal AccuScan GOLD reader, dispensing pump or graduated cylinder, filter syringes or Whatman No. 1 filter papers, sample dilution tubes with caps, a 100 microliter pipetter, a 500 or 1000 microliter pipetter and tips, and distilled or deionized water. Remember to always store test strips in the Stay Dry tube at room temperature. Do not freeze the test strips. Treat all used liquids and labware as if contaminated with DON. Neogen recommends gloves and other protective apparel, although they're not required during testing. Be sure to follow your company's policies. Avoid cross-contamination by using clean extraction supplies for each sample. Neogen's AccuScan GOLD system consists of a lateral flow test reader and data management software that interprets Neogen's Reveal Q test strips for mycotoxins. The system provides an easy method to objectively read, store, and analyze results. The AccuScan GOLD reader must be charged before use. Charging takes approximately 4 hours. The reader can be charged by attaching the power cord and the USB cable directly to the reader, or attaching the cord and the cable to the cradle and inserting the reader into the cradle. Once charged, the reader's power should last up to 3 days of regular use and up to 2 weeks with no use. To turn on the reader, push the button on the left-hand side below the screen. The main menu screen is used to access all sample testing, sample results, user identification, and other test support utilities. Prior to running a sample, you must enter the lot-specific QR code. To do this, place the disposable QR code contained in the kit into the QR code cartridge. Select the Scan QR code icon from the home screen and place the QR code cartridge in the reader. To scan the code, touch Run Scan. The specific information is automatically downloaded into the reader. Alternatively, you can manually enter the QR's 39-digit code. For instructions on how to do this, see the AccuScan GOLD user manual. It is important to scan the QR code for each new kit lot. To prepare the sample, begin by gathering all of the materials required for the test. The sample to be tested should be collected according to accepted sampling techniques. See the GIPSA sampling protocol or contact your Neogen representative. Obtain a representative sample by grinding a minimum of 100 grams of sample so that at least 75% of the ground material passes through a 20 mesh sieve. The particle size should be about the same as instant coffee. The sample is extracted with a ratio of 1 part sample to 10 parts distilled or deionized water. For this example, we'll use 10 grams of ground wheat with 100 milliliters of distilled water. The USDA GIPSA approved method uses 50 grams with 500 milliliters of distilled or deionized water. Vigorously shake by hand or mechanical means for 3 minutes. Or you can blend the sample for 1 minute. Alternatively, you can pipette 1 milliliter of the sample into a 1.5 milliliter microcentrifuge tube and centrifuge for 1 minute using a microcentrifuge. Allow the sample to settle and filter with a filter syringe or Whatman No. 1 filter paper. For each sample you're testing, place sample dilution tubes and Reveal sample cups in a sample cup rack. Add 1,000 microliters of sample diluent into each red sample dilution cup. Then add 100 microliters of the sample extract. Mix up and down with the pipette at least 5 times. Transfer 100 microliters diluted extract to a Reveal sample cup. Use a new pipette tip for each sample. It is important to make sure the pipette tip is fully charged with liquid and doesn't have any air bubbles prior to dispensing the liquid into the cup. Place a Reveal Q for DON test strip into each sample cup with the sample end down. Allow the strip to develop in the sample cup for 3 minutes. During this time, prepare the AccuScan GOLD reader to read your results when the incubation is complete. To do this, select the test category, Micotoksin Q+, using the category drop-down menu. Select the DON Q+ test type using the test name drop-down menu. Next, select the appropriate username under the username drop-down menu. Select Run Test once the test information is selected. A window appears asking the user to input a sample ID. Enter the ID and press OK. If you do not want to add the sample ID, simply press OK while leaving the field blank. Following the 3-minute incubation, remove the strip and insert it into the R-labeled strip cartridge. Fully insert the cartridge containing the strip into the reader upside down, sample port end first. When correctly inserting the cartridge, the test lines face downward into the reader. The reader's green light glows when a cartridge is inserted. The reader automatically begins analyzing the cartridge. Within 2 to 4 seconds, the green light turns off and 1 beep indicates the completion of the analysis. Removing the cartridge prior to completion can result in invalid readings. It's important to note that readings must be conducted within 1 minute of the completion of the 3-minute incubation. Readings conducted after 4 minutes may be inaccurate due to overdevelopment of the device. Strips must be read using the Reveal AccuScan GOLD reader. A results screen appears. Images of the test and control line zone appear on the center of the screen. The sample ID and result appears below along with the line intensities. To see the full test image, select View Image at the bottom of the screen. The test results are automatically saved. To reread the same strip, select Retest. To test a new sample, remove the cartridge and select New Sample ID. Input the new sample name and insert the new test strip in the cartridge into the reader. The test category, type, and user remain the same as the previous test, unless the user returns to the main screen to edit these fields. To transfer the results to the AccuScan GOLD Data Manager program, insert the reader into the cradle or attach the USB cable to the reader and then to a computer. Open the AccuScan GOLD Data Manager software on the computer. Select the sync icon on the software to pull data from the reader to the computer. If you have any questions regarding the use of the AccuScan GOLD reader, please contact Neogen. Manuals are also available. Neogen now has a full line of Reveal Q+ test kits, including aflatoxin, aflatoxin green, DON, zearalenone, fumonisin, ochratoxin, and T2-HT2. We have kit inserts and step-by-step pictured procedure brochures for all Reveal Q+ kits. Please contact Neogen for more details on our full line of Q+ test kits.